Muy buenos días. He recebido indicaciones de que la discusión será esencialmente hecha en inglés. Quería solo saludar al candidato, a la audiencia, sus compañeros y su familia. I'll myself inform on the discussion of your PhD thesis. This is the PhD thesis submitted by Henrique Nemesio entitled Biophysical characterization of membrane-active regions of structural and non-structural proteins from dengue virus. Myself, unknowingly, I'd like to thank the thesis supervisor, Professor José Villalaín, and the rectorate, and I'm honored for sharing this session. And I'd like to introduce you the other two jury members. On my right, Professor Selena Corabán García de la Universidad de Murcia y Jesús Salgado Bonito de Valencia. And on my left, Jesús Salgado. Further right, Pablo Castaminza from the CNB de Madrid. And on my further left, the jury secretariat, Professora Maria Amparo Esteves Pérez, aqui da Universidade de Alger. And I will invite the candidate to make a presentation of your PhD thesis and congratulate you for finishing just arriving to this final place.
Thank you very much. I will then start with a presentation of the thesis presented already, with research conducted under the guidance of Dr. Villalaín and Boullón here at the Cellular and Cellular Molecular Biology Institute. I shall divide this presentation into 5 distinct points. At first, introduction, followed by methodology, objectives, publications, and the final point of conclusions. Dengue virus is transmitted by a mosquito vector, and infections, people affected by this disease develop distinct symptomatology. For instance, dengue fever, or in more severe cases, dengue hemorrhagic fever, dengue shock syndrome. This virus is part of the Flaviviridae family and the Flavivirus genus. It has 4 distinct serotypes, and the virus itself is composed of a nucleocapsid enclosed by a host-derived lipid bilayer, where some 2 of the structural proteins are embedded. The immature particle here on the left is slightly larger than the mature particle on the right, and it has a characteristic spiky conformation. It is a serious public health problem. It affects mainly, it is endemic to the tropics and subtropics regions from 40 degrees north to 32 degrees south, and recently some cases in Madeira, Portugal, Nice, France, and a peninsula in Croatia, some cases have been reported, thus increasing the risk or the severity and the need to address a cure for this virus. Its life cycle, as many flaviviruses, it enters the cell by receptor-mediated endocytosis. It has not been defined a specific receptor for this virus, and on the contrary, several have been defined. And following endocytosis, a decreasing pH inside the endosomes triggers the fusion process that results in the release of the nucleocapsid into the cytoplasm of the cell. After the dissociation, then the polyprotein is then translated on endoplasmic reticulum membranes, and all of the remaining processes, such as viral replication and virion budding, also occur in endoplasmic reticulum-derived membranes. Following virion assembly, this particle then starts its transit through the trans-Golgi network, and it's in this, during this transit that the final step of maturation occurs, the cleavage of the PR peptide by furin. Following nucleocytosis, the PR peptide is then released, and the particle is rendered fully mature. This virus, it has a viral RNA genome. It is a positive strand of RNA that contains a single opening frame encoding a single polyprotein of about 3,000 amino acids. This protein, after appropriate cleavage and maturation by viral and host proteases, gives rise to 3 structural proteins and 7 non-structural ones. It is important to note that this virus, in all of its steps of the viral cycle, and on all of its steps of the viral cycle, it is always associated with lipid membranes, and they have quite distinct lipid composition. Now, focusing on the proteins considered for this thesis, the capsid protein, it is composed of 4 alpha helices interconnected by short loops. As you can see here, this is the monomer of the capsid protein, and it readily forms dimers in solution. This is the dimer in 2 different views that contains 2 distinct regions. A highly hydrophobic region composed of helices alpha 2 from both monomers, and a region with a high concentration of positive charges formed by helices alpha 4 of both monomers. As for functions, this protein is responsible for the specific encapsulation of the genome, and it has been found to accumulate in lipid droplets and localize to endoplasmic reticular membranes. For the PRM protein, it is composed of 2 main regions, a mainly alpha helical stem region at its N-terminal region, and a C-terminal region composed of an alpha helical stem region, followed by 2 transmembrane helices. Now, it has been long known that one of the functions of this protein is to prevent an early fusion process by burying the fusion loop containing the protein inside itself. This is a dimer of these 2 proteins that's found at the surface of the virion. Recently, it has been found that the M protein triggers apoptosis. It interacts with proteins from the transportation machinery, and it also interacts with ATPases. Thus, some authors postulate that it might have a role in the pH regulation inside endosomes, and have some role in viral fusion processes. For the protein, it is a class 2 fusion protein composed of 3 mainly beta-stranded domains. The N-terminal domain 1, shown here in red, is flanked on the one side by domain 2, where the fusion peptide is located at its tip, and on the other side by domain 3, where the putative receptor binding sites are located. Now, the fusion process itself relies heavily on this protein, and it occurs in 2 steps. A decrease in the pH inside the endosome triggers a conformational change where domain 2 protrudes away from the membrane, the viral membrane, and the fusion peptide inserts itself into the host membrane. This is then followed by a folding of domain 3 in the direction of domain 2, approaching both membranes, viral and host membranes, that ultimately results in a fusion. The C-terminal part of this protein is composed of 2 amphipatic helices, EH1 and EH2, that are involved in the viral assembly. And these are followed by 2 transmembrane domains, TM1 and TM2, involved in membrane fusion processes. As for the NS4A protein, the following 3 I shall describe are highly hydrophobic proteins, and all co-localized to the replication complex of this virus. This one, NS4A, is composed of at least 2 transmembrane segments, separated by a membrane interacting domain, and followed by a C-terminal 2K fragment. This fragment is required for the translocation of the following protein, NS4B, into the endoplasmic reticulum. As for functions, the N-terminal region of this protein affects the ATPase function of NS3. It co-localizes, as I said, with the replication complex. It's required for the antagonism of the interferon response, and it upregulates autophagy processes that ultimately result in a prolonged cellular lifespan. This protein is also found, as it co-localizes with the replication complex, and in the plasmic reticulum-derived membranes. As for the NS4B protein, it is composed of at least 3 transmembrane domains and 2 hydrophobic segments at its N-terminal region. This protein is the chief antagonist of the interferon response, and it also inhibits the host RNA interference response. It modulates the helicase function of NS3, and it has also been described recently that it forms dimers, and it is this region between these 2 transmembrane domains that is postulated to be the monomer-to-monomer contact. As for the NS2A protein, it is also, as I said, highly hydrophobic, composed of at least 5 transmembrane segments and 2 membrane interacting domains. It also co-localizes with the viral replication complex. It is required for the proper processing of NS1, and with the previous two, it also inhibits the interferon response. There are, in general, we could delineate at least 3 different control and management strategies for the control of this virus, both its spread and infection. One, at the first level, is the control of the mosquito population. Some studies refer to the use of sterile males, flightless females, and even mosquito parasites. Another one, passive, of course, through the management of the symptomatology associated with this disease to prevent serious complications. And the final one is the development of antiviral drugs and vaccines. There is still no effective cure for this virus nowadays. Therefore, the countries, especially those countries where this virus is endemic, follow WHO guidelines that rely heavily on the first 2 topics. Now, some targets have been devised or proposed by several authors recently, and also strategies to counter this virus. At first, the development of antiviral drugs. Several targets, especially, they are designed to target the conformational changes needed for the proper viral replication. For instance, there is a binding pocket between domains 1 and 2 of E that could be prevented. A hinge motion, this domain does a hinge motion to result in a fusion process. And also, some peptides have been found to target specific regions in the variant, the mature variant, and inhibit the proper formation of variant, genome-like variants. There's another drug, salgosivir, it's a glycosylation inhibitor, and it has been found to efficiently inhibit the glycosylation of NS1 and this results in a 95% of viral inhibition. As for vaccines, there are 2 main obstacles to the development of vaccines. One is the need to address the tetravalent nature of this virus, so that there is no cross-reactivity. And another one is a phenomenon called antibody-dependent enhancement of dengue. This happens when people are infected by a secondary heterologous subtype, serotype, and they develop a much more severe response to dengue. Now, moving on to point number 2 of this thesis, the methodology used in this work, just a short synthesis. We use peptide libraries and membrane model systems to represent both regions in the native proteins on the one hand, and on the other hand, cellular membranes. And we characterize the interactions between these two by at first representing the water-to-membrane and water-to-interface free transfer energies of all residues. This methodology, I shall explain in the next slide, allows us to highlight possible transmembrane domains, membrane-interacting domains, and protein-protein interacting regions. We also want to assess the effect this peptide had on lipids by studying the thermotropic behavior of lipids in the presence of this peptide, by measuring steady-state fluorescence anisotropy of VPH and its derivatives. We also assess the membrane rupture these peptides elicit on membranes, and the binding of some of these peptides to membranes using an indirect way, using a pH-sensitive fluorescent probe. We also assess the effect of these peptides on the thermal behavior of lipids by differential scanning calorimetry, and the overall secondary structure of some of these peptides in the presence of membranes was assessed by infrared spectroscopy. Now, as I said before, the representation of the water-to-membrane and water-to-interface free transfer energies. As you can see here, we project a 3D alpha helix, the known structure of the alpha helix, to 2D. As you can see here, these are all of the terms of the alpha helix, and then we represent for each residue its value in a given scale. This is just an example of that situation. And we can see with this representation, of course, we assume at first that it is an alpha helical representation, but at first it's just for the sake of visualization. We have tried it with several proteins to try and see if this would work, this methodology. It does work, it matches pretty well with known segments, and this allows us to see patches inside in the sequence of the protein that might be, as I said, membrane interacting or protein-protein interacting. Moving on to the objectives, the main objectives of this thesis. There were, at first there was one main objective, the biophysical characterization of membrane-active regions of structural proteins and non-structural proteins from dengue virus. And from this first objective, I'm using the methodology I already described. This objective resulted in the publication of 2 scientific articles, and other 2 that were sent for the publication. One of them had B5, we just knew today that it was already accepted. Now, from this first objective stemmed another one, the biophysical characterization of a peptide derived from the capsid protein of this virus that had dramatic, we had seen, as I will show, that had dramatic effects on membranes, and it resulted in the publication of a scientific article. Now, Now, presenting a short summary of all the results obtained in each of these publications. First publication, the membrane-active regions of the dengue virus proteins C and E. As I said, we used peptide libraries encompassing the full length of these proteins, and these peptides have a characteristic. It's that 2 and 3 consecutive peptides overlap between themselves. So one might think not as a given peptide, but regions in the protein. These are at first the first results we obtained. The leakage measurements, where we have here on the y-axis, we have the percentage of leakage with respect to control. And each of these columns represent one of those peptides in the peptide library and the residue number in the protein. And we also represented, then as you can see here, we detected at least one peptide that had dramatic effects on rupture of membranes. And these are all distinct lipid compositions. As you can see, it has no specificity, this effect on the lipid composition of the membrane. And now here on the left, as I said, we represent several scales of hydrophobic moments, hydrophobicity and interfaciality using that representation. And as you can see, there was this region that was highly hydrophobic and with high propensity to be with interfaciality. And this is the charge density using the same representation. As you can see, this matches, this highly coincident with a highly hydrophobic region and that had leakage effect. And it is also coincident, this region, with the predicted or the suggested hydrophobic region in the structure of the diamond. As for protein E, we could detect, using all of this methodology, at first with leakage, 5 distinct regions that had any effect, sometimes significant, on the rupture of membranes. And using the same representation, as you can see we have here, the first region is highly hydrophobic. It had high leakage. It was coincident with the fusion peptide of the E protein. As for the second and third regions, they were not so hydrophobic and they do not cover the full horizontal length of this representation. They had significant leakage and they are coincident with a protein-protein interacting region and with a beta structure that covers the hydrophobic pocket between these 2 domains, domain 1 and domain 2. And the last 2 detected regions are regions at least 4 and 5, are coincident. Region 4 with the EH1 helical region of the C-terminal part of this protein, and region 5 is coincident with the EH2 helix and the transmembrane domain 1. As you can see, once again, at least, especially the last part, they have no significant differences as to their effect on the rupture of membranes. Moving on to publication number 2, a hydrophobic segment of dengue virus C protein, interaction with all the membranes. As I said previously, as I have shown previously, we had seen that this peptide from this region had a significant, I'd say dramatic effect on membranes, and it was coincident with a high hydrophobic region. So we conducted a more thorough biophysical characterization of the interaction of this peptide with membranes. At first, as you can see, the sequence of this peptide, it is highly conserved among all serotypes of dengue virus, so it might be, at first one might say, it should be important for the virus. And first, as you can see here, we assess the binding of this peptide to membranes, but using FPE, a pH-sensitive fluorescent probe, because this peptide, as you can see, has no tryptophan. So we could see that it did bind to membranes significantly, but more so to membranes composed of negative lipids that are shown here, for the sake of simplicity. And we then assess the effect these peptides had on the leakage this peptide elicited on several membranes of distinctive composition. As you can see, this is the caption, but these are all membranes containing negative lipids. And this peptide affects much less these peptides, well, at least at over 300 to 1 lipid to peptide ratio. And in some cases, as you can see, those membranes containing either sphingomyelin or cholesterol, and in general, the membrane is neutral. As you can see, even at 800 to 1 lipid to peptide ratio, this peptide elicited over 80% leakage. So it is without a doubt that it does affect membranes in some way. So we wanted to characterize it further. We resorted to the use of other two fluorescent probes, dextran derivatized, fluorescence derivatized, and to assess if this peptide could induce formation of pores in the membrane. Why? And if so, what would be the size of those pores? As you can see, A and B are two distinct dextrans. This is a larger one. They have no significant differences. This peptide elicits leakage at the addressed lipid to peptide ratio. So one might say if this peptide induces pores in the membrane, they are quite large. And we also assess the effect of this peptide on membranes by measuring the steady-state fluorescence anisotropy of DPH located, that's distributed to the inner core of the membrane, and the derivative of this probe, TMA-DPH, that is located closer to the surface of the membrane. We assess different lipids, but the most significant effect, or the most significant fact in this case, result, is that this peptide affected more the DPH probe than the TMA-DPH probe. So one might suggest that it has an effect more at the deeper level than at the surface. These results were mainly seen by a reduction in the cooperativity of the transition of each of these lipids and an increase of the post-transition fluorescence anisotropy. These results were confirmed by differential scanning calorimetry. I mean, the effect this peptide has on some lipids. There was a decrease in cooperativity seen here by the broadening of the main transition peak of several lipids except the BE, and in some cases it even shifted the transition temperature. Now, assessing the overall secondary structure of this peptide in membranes, as you can see at first, in solution, this peptide has an overall beta aggregated structure. Wow. These two peaks at 1624 and 1694 are representative, at least in the literature, of beta aggregated structures. There was an effect is that at approximately 40 degrees Celsius, there was a shift of this peak to this other peak at 1684 centimeters that's associated with beta terns. So as this is clearly seen here, at approximately 40 degrees Celsius, there is that shift. So, and then when we inserted this peptide in the presence of membranes, in DNPC membranes, this peptide affected slightly the transition temperature of DNPC, and it changed dramatically. Its overall secondary structure was changed dramatically in the presence of this lipid to mainly alpha helical and unmodular structures, as you can clearly see here. As for the MPG membrane, as you can see by the carbonyl head, there is these two bands that are usually overlapping in the pure lipid are separated. We can clearly see a small shoulder here, meaning that the peptide effect somehow these are associated with those carbonyl heads that are interacting. They are hydrogen bonding, are involved in hydrogen bonding, and the other band with those that are not involved in hydrogen bonding. So the peptide does have an effect on the carbonyl head of this lipid. And furthermore, as you can see here, the secondary structure of the peptide is quite similar to that in solution, but that same effect at 40 degrees Celsius, that shift of those two peaks, is not seen in this case. So one might assume that this peptide has a fixed structure in the presence of this lipid. The same happens with BMP, where it affects the carbonyl head of the carbonyl band of this lipid, and its overall secondary structure is maintained, yet there is a small shift in those two peaks. Moving on to publication number 3, NS4A and NS4B proteins from dengue virus, membranotropic regions. And once again, the first experiment is always a screening of those possible regions that elicit any leakage to membranes. We used these now are here described synthetic lipids, and some of them are naturally extracted. And as you can see, at first, the most striking is that we can define at least 3 different regions in the NS4A protein. As you can see here, 1, 2, and 3 to 2 here fragment. And in the NS4B protein, we have 5 distinct regions seen here. And the other noteworthy result is that there are no, at least the most significant leakage-inducing regions, there are no significant differences with respect to lipid composition, once again. Now we also assess the effect these peptides have on the thermotropic behavior of DMPC membranes. Some peptides shifted the main transition of the lipid, the temperature of the main transition, and other peptides induced an increase of the post-transition fluorescence anisotropy. This is the DPH probe, once again, located at a deeper core. The same results were obtained from NS4B. Some peptides shifted the temperature of the main transition, and other peptides affected the fluorescence anisotropy at a post-transition level. Now, just for the sake, because these were a lot of results, so I tried to synthesize it a little bit more, and as you can see here for the NS4A protein, we conducted those studies, once again, of hydrophobic patches in the bidimensional representation, and we could detect 4 distinct regions. As you can see here in the caption, in green are the results from those bidimensional bands. These are coincident with the leakage results, as you can see here, more or less coincident, and also coincident with anisotropy experiments, but to a lesser extent. And interestingly, all of these regions detected that we could define and highlight are coincident with the predicted or the suggested topological model of this protein, shown here in blue. These are those regions. So, and for the NS4B protein, the matching was not so easily seen, well, at least for some of those patches, and the topological model for the N-terminal region is quite spread. It's quite widespread. So, but we could also detect in this NS4B protein several hydrophobic patches or protein-protein interacting regions, and also leakage-inducing regions and anisotropy results. But the ones that match rather well are the transmembrane domains. That, as you can see here, they are all matching in all experiments. And interestingly, there was another one, was that we could detect by that bidimensional presentation a possible protein-protein interacting region. As you can see, it is this region. And it was, we suggested it might be protein-protein interacting, and we could see that experiments developed or designed to detect membrane interacting regions, it came back negative in this case. And interestingly, this region is coincident with the monomer-to-monomer contact, the suggested monomer-to-monomer contact. So it is quite clear this membrane, this protein, once again, these 2 proteins have quite a significant effect on membranes. Moving on to publication number 4, membranotropic regions of dengue virus, PRN protein. This peptide was sent for its publication, but it was still not accepted. We had no news. So in this case, at first, the assessment of the membrane leakage induced by these peptides of this protein, we used different lipid compositions. We could detect at least 2 different regions. One located in the PR moiety, and another one coincident with the transmembrane regions of this M protein. I would like to stress now, in this case, that this region, right previous to this C-terminal region, is coincident with the stem region of the M protein. Just as I will show later on. We assessed the effect these peptides had on the thermal behavior of 2 lipids, BAPC and BAPG, And here we have the representation of the thermograms of lipid, of pure lipid, and lipid in the presence of each of those peptides. As you can see, there was no peptide that affected significantly, well, no significant effects are seen on the DMPC membranes, but in the case of the DMPG membranes, we have peptides that reduce the cooperativity of the transition. Some peptides induce the formation of mixed phases that could be enriched and impoverished in peptide. And there was, as I said before, this region of the stem region that had no leakage effect whatsoever. In this case, it did decrease significantly the cooperativity of the transition of DMPG. We also saw the effect, but mainly the C-terminal region of this protein had a significant effect on the DMPC membranes, on the anisotropy of DPH inserted into the DMPC membranes. It shifted and shifted the transition, main transition temperature. It decreased the cooperativity of the transition, and it increased the post-transition anisotropy. Now, as a summary, here we have the representation, the bidimensional representation for this protein. As you can see, there are distinct regions, but the C-terminal ones are the ones that are the most hydrophobic. And trying to show all together, we have a first region that was mildly hydrophobic, that had some effect on the leakage of membranes, of fluorescent probes and membranes. And the second region, this green region, is coincident with this same green region of contact between the PR peptide, PR moiety, and the protein, and it did not induce significant leakage. This is followed by this region, this blue region, coincident with the C-terminal region of the stem part, stem moiety. It had no significant leakage effect, but it did affect the calorimetry, the thermal behavior of the DMPG membranes. As for the C-terminal region, it was highly hydrophobic, it did induce high leakage, and it also affected significantly the thermotropic behavior and the thermal behavior of lipids. For the fifth and final publication, membrane interacting regions of dengue virus NS2A protein. This one was the one that we knew today that was accepted. At first we conducted the same experiments, but with this NS2A protein. These are summarized in this slide. As you can see, we could detect, using the same again, several lipid compositions, we could detect at least two different regions. One coincident with peptide 5 that had a selective effect on leakage depending on the lipid composition, as you can clearly see, clearly observed in this case, and a broad region that is coincident with the transmembrane segments of this protein. Here, it's slightly different, but the same results, the same scales of hydrophobicity and interfaciality are shown here, but using in a linear scale in this case. This region, we could detect at least 10 different regions, but this region, coincident with this peptide, has some characteristics that are interesting. That the hydrophobicity shown as the solid line and interfaciality shown as the dotted line are overlapping, are high. It has hydrophobicity and high interfaciality, and they are overlapping. These are seen several several segments of viral proteins that are at the interface and interact with the interface have this characteristic. Interestingly, it was coincident with this, the first membrane interacting domain of this region. So we selected this peptide to carry further biophysical characterization. At first, using the same methodology used before for the peptide derived from dengue capsid protein, this peptide had no tryptophan also, so we had to use that pH-sensitive probe. And summarized here, as you can see, these are membranes composed of negative lipids, with a large proportion of negative lipids, and these are overall neutral membranes, neutralionic lipids. As you can see, it binds more to a higher extent to those composed of negative lipids. And interestingly, in this case, this is an inset where we have, as you can see, this is the fluorescence, well, the binding, the measure of the binding of this peptide to this membrane, to this probe, and with respect to the proportion of negative lipids in the membrane. There is a linear relationship between the binding of this peptide to membrane and the proportion of negative lipids in that membrane. The same separation of groups of negative lipids and zwitterionic lipids was seen by leakage experiments. You can see this peptide affected to a higher extent lipids composed of negative, membranes composed of negative lipids than membranes which are composed of zwitterionic lipids. So we wanted to assess if this peptide has a selective effect to negative lipids. Could this have a major electrostatic contribution? So we assessed the effect of this peptide on DNPG membranes at distinct ionic strengths of the buffer and at different ions. As you can see, well, we can appreciate at first in this case that there is a reversal of the curve of the anisotropy of the peptide, the lipid in the presence of the peptide, reversal to the pure lipid. It approaches the pure lipid. There is a decreased effect. This is seen here just to be represented in this case, the slope at the transition temperature and difference in anisotropy at a post-transition level. As you can see in this case, in black, the filled symbols are the pure lipid, and the empty symbols are added in the presence of the peptide. You can see there is an increase. This is the modulus of the slope, and there is an increase of that modulus. Once again, there's a reversal to the pure lipid, but of course, even at concentrations as high as 300 millimolar, it still has a significant effect on membranes. This is also seen in the case of the difference of the anisotropy between pure lipid and pure lipid and peptide at a post-transitional level, and there is a decrease of this difference approaching zero with increasing salt concentration. So this significant effect has a major electrostatic contribution. This we also assessed by differential scanning calorimetry. As you can see, DNPC membranes at either concentration, the peptide did not elicit any significant effects. And as for negative lipids, the peptide at low salt concentrations affected significantly the thermal behavior of these lipids, either by reducing the conductivity, by the induction of mixed phases, and the shifting of the transition temperature. And on the other hand, with increasing salt concentration, this effect is decreased. In the case of DNPS and DNP, at 100 millimolar, it is almost similar, completely similar to pure lipid, and in the case of DNPG, the cooperativity is increasing, yet it still does not reach the final pure lipid. So once again, it has a major electrostatic contribution. This effect was also seen in the case of infrared spectroscopy experiments, but at first, as you can see, this is the secondary structure of this peptide in the presence of DNPC membranes. At low, these A and C are low concentration of salt, and these are the high concentration. At either concentration, the overall secondary structure of the peptide is quite similar. Overall, aggregated structures with contribution of mainly of alpha helical and unordered structures. This is clearly seen here with this insect. Another effect was that at low salt concentrations, this peptide shifted the maxima of the carbonyl band of this lipid to higher frequencies. They are associated with a differential increase of those two I referred previously. The carbonyl heads that are involved in hydrogen bonding and those that are not involved in hydrogen bonding. So, and in the case of DNPG, we clearly see a significant effect. At low salt concentrations, the transition is completely abolished. It's severely decreased the cooperativity transition at high concentrations. And another aspect is that at low concentrations, this peptide changed its overall secondary structure to mainly alpha helical, shown here by this figure, mainly alpha helical and unordered structures. So this peptide has a significant effect on membranes, but it has also a major electrostatic contribution. Now, as a summary, we highlighted the membrane active regions of several of dengue proteins, In the C protein, we detected a region that had a dramatic effect on membranes, independently of lipid composition. For the PRM protein, we could detect at least 4 different regions with membrane interacting capabilities seen in different experiments. For the A protein, there were 5 of these regions. For the NS2A protein, there were 2 regions, one with composition-dependent effect, leakage effect, and another one, a broad region coincident with the transmembrane regions of domains of this protein. For the NS4A protein, we could detect at least 3 different regions, and for the NS4B protein, at least 5 different regions. And once again, for these 2 regions, the matching with the proposed topological model is quite significant. Now, with the permission of the evaluation committee, I shall now read the conclusions of this thesis. Point number 1. Relying on an array of several biophysical techniques, we highlighted different membrane-active regions in all structural proteins of dengue virus, and its less characterized non-structural proteins. Number 2. At least 5 highly hydrophobic regions between proteins C and E could induce the rupture of model membrane resistance to a large extension and coincided with regions deemed essential for some of those proteins' functions. Point number 3. One of those highly hydrophobic regions, from residues 39 to 56 of protein C, matching a putative membrane interacting domain, binds strongly to membranes with a slight preference for membranes with anionic lipids. It induces a virtually complete membrane rupture independent of composition and modulates thermotrophic lipid phase behavior. We suggest this region might be involved directly in the membrane fusion process with the E protein. Point number 4. We have found at least 5 membrane-active regions in NS4A, 3 of which were the transmembrane segment and other 2 in close association with it. Fragment 2K did not induce rupture of ER2 membranes, suggesting that it either does not interact strongly with said membranes during the viral cycle, or that it is inserted into them in the replication complex. Point number 5. Both membrane traversal regions and membrane interacting regions were defined in NS4B, suggesting that this protein is involved in membrane rearrangements, protein-lipid, and protein-protein interactions. Point number 6. Other than the 2 transmembrane regions, the PRM protein presented 1 protein-interacting region, possibly the region of interaction with E, a pre-transmembrane segment matching the stem region with significant effects on membranes that could switch between membrane-bound and unbound conformations, altering the membrane structure and polymorphism. Point number 7. We have identified a broad region in NS2A where membrane rupture capabilities independent of membrane composition and the membrane interacting region with composition-dependent effects. Point number 8. A region encompassing residues 30 to 55 of NS2A called dense 25, where composition-dependent effects on membranes bind strongly to membranes with anionic lipids and exerts modification to the structure of anionic membranes with a large electrostatic contribution, effect reduced in the presence of cholesterol. We propose this region undergoes conformational changes depending on the local concentration of anionic lipids that render it membrane-active or membrane-inactive, contributing to the viral replication. Thank you very much for your attention.
Thank you very much. And before proceeding the discussion with the jury members, I would invite any doctor in the audience, if you'd like to make questions to the candidate. This can be also carried out in Spanish.